everyone, welcome to the next episode of my science podcast. Today I'm really delighted to welcome Professor Grant Jensen, who is a Professor of Biophysics and Biology at Caltech, whose work focuses on using electron cryotomography to study cells and viruses. His projects range from theoretical studies on the mathematics of three-dimensional reconstructions to direct imaging of individual protein complexes to tomography of viruses and cells. So thank you so much for joining me today.
Happy to be here, Martha.
So to begin with, may you tell us about your career so far and how you got to where you are today?
Okay, thanks for the question. I was born in Los Alamos, New Mexico, and some people have heard of that town, but not everybody. Basically, in World War II, the United States recognized that we might be able to tap nuclear energy to develop a weapon, a nuclear weapon. And in order to do it, they picked a remote site of the Western United States, in New Mexico, where they could do this work in secrecy. And that was the birth of Los Alamos National Lab. And then after the war, of course, they succeeded. Then after the war, everyone realized that it would be important for the democracies of the world to stay far ahead of their adversaries in nuclear weapons technology, and that we also might be able to use nuclear energy, tap the same kinds of processes to produce a great source of energy. So they kept all the scientists that they could in Los Alamos. Anyway, progressed another 22 years, 25 years, and they recruited my dad to be there as a physical chemist, and that's where I was born. So Los Alamos is a really strange town because it has the highest concentration of PhDs of any county in the United States. And growing up there, I mean, almost everybody that was in the workforce was a physicist. So I was pretty good at school, so I figured I'd be a physicist too. Everybody was a physicist. Anyway, so when I graduated, I went to Brigham Young University in Provo, Utah. It's a religious school run by the Church of Jesus Christ of Latter-day Saints. I'm a member. I wanted to go there, and so I went and I majored in physics. And when it came time to pick a project for a senior thesis, some of my friends were picking things related to astronomy, and I wanted to do something a little bit more relevant to the human condition. And my attention was captured by these posters on the wall of big macromolecules and advertisements for graduate schools in biophysics. And I fell in love with that and loved the practical applicability of understanding protein structures and the potential drug development that would arise from that. So after graduating in physics, I went on to do biophysics at Stanford. And I worked with this tremendous scientist named Roger Kornberg, who ultimately won the Nobel Prize for determining the structure of RNA polymerase, the enzyme that reads DNA. And I joined his group because it was an evening, pizza night where all of the incoming graduate students got together each, some night, each evening, each week, one particular evening. And they would have pizza, and a professor or two would tell us something about their research. And this would help us pick where we wanted to do rotations. Roger Kornberg got up and he showed us pictures of RNA polymerase, this enzyme. And he taught us about how it can read DNA, make a copy of it. If it makes a mistake, it can proofread, go backwards, fix it. It's regulated by all these different processes. He said it's the most important enzyme there is. I'm convinced that's true. He was right. And I realized, and so he was taking pictures of this, his group was, with an electron microscope. And I realized it was that perfect union of things that I would love. I love physics, and the electron microscope is like the most glorious. There's a famous experiment in physics, the two-slit experiment. We don't have time to talk about it. But the electron microscope is one of the most glorious physics experiments every time it takes a picture. And so that interested me. Also, I knew that this field would require a lot of mathematics and computer science to understand the images and process the images. It was a developing field, so we need to write new software that dealt with 3D objects and math. That was a love of mine. And all of this work would be done in the service of understanding the most important enzyme out there, RNA polymerase, or everyone can argue what they think is the most important enzyme, but all in the service of understanding how cells work. And I just thought I'd died and gone to heaven. So that night, I called Roger and said, I'm done. I don't want to do any rotations. I just want to join your group. And he was taken back because that's not normal. You're supposed to do 3 rotations and then pick your favorite lab. And I didn't want to do any of that. I just wanted to do electron microscopy, write software, do math in the service of understanding cells. And Stanford made exceptions and let me do it. So I joined Roger's lab and I learned electron microscopy of biological molecules. Then I did a postdoc. One of the fathers of this field, cryo-EM, cryo-electron microscopy, which is the kind of electron microscopy that's used to image biological macromolecules. One of the fathers of this field was across the bay in Berkeley. His name is Kenneth Downing, and he's a great postdoc mentor. So I joined his lab to do my postdoc. And while I was there, I heard a seminar from Professor Wolfgang Baumeister, who showed how you could take pictures of cells and take lots of pictures of a single cell while you rotated the cell around. And by doing that, you could build up a 3-dimensional understanding of everything inside the cell. I saw that, I left the seminar, I went back to Ken in the lab, and I said, that's it. This is what I'm doing. I'm going to learn how to do this, and I'm going to apply it to understand what's inside cells. And he generously supported me in that direction. I started visiting Professor Baumeister's lab, and the people in that lab taught me how to do it. And just then, Caltech, California Institute of Technology in Los Angeles, had also recognized the potential of cryo-EM and had opened a faculty position with interest in cryo-EM. And they were kind enough to, take a chance on me and give me a job. And we bought a great cryo-EM, and my group has been there taking these 3D pictures of cells for about 18 years, and it's just been super fun. Then I guess just to finish the story, just last November, my alma mater, Brigham Young University, asked me if I would come back and be the dean of their College of Physical and Mathematical Sciences. And it was a tough decision, because it's more administrative than research. But I love what BYU is trying to do. I wanted to give back. And so I've made that transition. So right now I'm the dean of a college at BYU. And I Zoom with the people that are still in my lab at Caltech, and we're still doing some cool science. But the plan is that that group at Caltech will diminish gradually, and I'll build another group with a smaller group with less ambitious goals here at BYU while I'm the dean. All right, long answer.
Amazing. No, it's fascinating. It's wonderful. So it's a relatively new field, I guess. And so may you tell us about how you've described a new cryogen mixture that facilitates plunge freezing? I mean, you've done so many things in this field. You've done that. You've shown other important things like that liquid nitrogen is actually a better coolant than liquid helium. You've characterized the first stage for routine dual axis tilting, automated sequential tilt series acquisition, developed a database for the storage of the tomograms, and you developed a new method for denoising. You've done all this amazing stuff. So can you tell us a little bit about some of those things?
Sure, I'm happy to. Let me keep it pretty basic and put it in a bigger context, if you don't mind.
Mm.
I want everyone to think about how much it advanced biology when light microscopes were invented, and all of a sudden scientists could see bacteria and these little cells that could swim around, and they saw that all tissues were really based on cells. Then microscopes got better, light microscopes got better, and eventually fluorescent markers were discovered and refined. These are little fluorescent molecules that you could put on something inside of a cell to see where that element of the cell was with a fluorescent signal. And this, has advanced biology so much. Well, then as people started to understand what electrons were, and that they had wave-like properties as well as particle-like properties, imagine how exciting it must have been for the first person who realized that you could build a microscope that used electrons rather than light. The reason that would be so exciting is that the resolution of a microscope in many senses is limited by the wavelength of radiation that it uses. So light, visible light, goes from about 400 nanometers to 700 nanometers. So you could see small bacteria, but it was hard to see inside. You could see the shape of a cell, but it's hard to see inside of a cell. Electrons, though, have wavelengths that are on the same order of magnitude as an atom. And so people thought, wow, if we could build a microscope that used electrons, we might be able to see things as small as atoms. And, so it's fascinating how this worked. Anyway, electron microscopes were developed. They worked. The earliest ones weren't good enough to see atoms, but today's electron microscopes are. So once the cell biologists heard about electron microscopes, they thought, wow, we got to look inside cells and see what's inside cells. But there was a number of challenges. One of the challenges is that the inside of an electron microscope has to be a very high vacuum, because if you have air in there, the electrons hit the air and they get scattered and absorbed, and so you can't have a microscope. You got to get all the air out. But you can't take a cell and stick it in the middle of a really high vacuum. The thing will violently boil. It would essentially explode, but the process would be boiling really. And so you have to figure out a way, how do I keep the cell intact while I can get it in this high vacuum? The first ideas were, what if we use chemicals to staple all of the molecules together so they can't get away from each other and explode? And these molecules are called crosslinkers or fixatives. So the idea was, what if we can wash the cell with fixatives that just kind of staple everything to whatever's next to it? Then we got to get rid of the water, because the water, if you put the water in a vacuum, it boils violently until it's gone. So much it's almost like an explosion. So what if we wash the water out and we flow into its place molecules that can become a plastic? Well, this is a resin. So wash in this resin, and then if you heat up the resin, it will harden into something that's like a block of plastic. And then we would have this hard plastic block of material that had the cell, all the macromolecules of the cell embedded in it. And that we could stick in the high vacuum of an electron microscope and take a picture of. Well, it turned out that the plastics, they're very similar in structure, in the atoms involved in the molecular structure, as the molecules that build up the cell. So it's really hard to see what's plastic and what's part of a cell. And so then stains were developed, stains like uranium or lead or osmium. People figured out how to wash these plastic preparations of cells with a stain, and the stain would stick to the biomolecules, like the lipids that show the membrane, or maybe they would stick to the proteins. And then you could see them in the midst of all the plastic. So this is the basis for what we would call traditional thin section EM, where you would take a cell, cross-link the molecules, exchange the water for plastic, cut it into thin sections, because another limitation of electron microscopes is the electrons can't go through very deep material. It has to be very thin. So you cut this plastic into sections, then you wash it with a stain like lead or osmium or uranium, and then you can take pictures of where the stain stuck. Okay, it sounds far removed from a cell, but the truth is it worked great. And if if you study cell biology and you get a textbook, page after page, you'll see a lot of pictures of the insides of cells that came from that traditional thin section EM method. If you go to medical school and take histology, it'll be page after page after page of these pictures. This is how you'll learn about what's inside cells. And a lot of what we know about the structure of cells came from this method. Okay, but there's a lot of problems. If you imagine if you were trying to look at a fish tank, that's like a cell. And the little fish in the tank are like the proteins that do everything. And imagine if you poured into the fish tank some glue, that every time a fish stuck to the side of the tank, it would get stuck there. And then if another fish hit that fish, it would stick to the first fish. That's what chemical fixatives are like. They just stick everything to whatever's next to it. But if you did that to a fish tank and then took all the water out, that's not exactly what the fish tank looked like. So other scientists had the idea, what if we could freeze a cell so that if it was cold enough, we could put it in the electron microscope in the high vacuum, and the thing wouldn't boil violently anymore, because there just wouldn't be enough energy for that to happen if it was really, really cold. And that's the essence of cryo electron microscopy, or cryo-EM. But if you just take a cell and cool it slowly, the water will form ice crystals. And everybody knows that ice crystals are less dense than liquid water. And so what that means is that the molecules expand. And so if you take a cell and slowly cool it down, most of the cells, the water will crystallize, expand, and break the cell open. It rips it apart. Also, the proteins inside of it will lose their shape, because they're chemically, they fold up into the shape they do when they're surrounded by liquid water. If you crystallize that water because you freeze it, the protein takes on a wrong shape. It falls apart. So an idea arose that what if we freeze cells so quickly that the energy to rearrange where the water molecules are gone, that energy is gone so fast that all the molecules will just stop where they are. We'll freeze it so fast that the water doesn't have time to form crystals. Everything just stops where it is. And we keep it that cold. Then we can put it in the vacuum of an electron microscope. And actually, one of my colleagues at Caltech for the last decade or more, a great friend of mine is named Alistair McDowell. And Alistair was a postdoc in the lab of Jacques Dubochet when this technique was first developed. And Jacques Dubochet ended up winning a Nobel Prize for that. And so it's been really fun to be close to Alistair as they celebrated his work with his colleagues in developing this method. Bottom line is they figured out how to do this. What you do is you take a cell and you drop it, you actually shoot it into a very cold liquid, and it freezes fast enough to prevent ice crystals. Mm. Okay, so then we've refined what it is, what liquid should we use. It's liquid nitrogen is very cold, like 186 degrees below 0 Celsius. And it's available. You know, most biochemistry labs have liquid nitrogen. It's not that expensive. You can get a bunch. So, but liquid nitrogen is not the best for freezing cells. And maybe I'm getting too technical, but it turns out, you know, liquid ethane and liquid propane are better at sucking heat out of the cell. So what we actually do today is we have a mixture of ethane and propane, liquid ethane, liquid propane, that's cooled by liquid nitrogen. And we drop the cell into the ethane and propane, and it freezes so fast that ice crystals don't form. And that's how we can stick it in the EM. And now when we look at it, there's no chemical fixatives locking everything into unnatural positions. There's no plastic in there causing all the molecules to change their shape. There's no stains, because we can actually see proteins directly because they're a little bit more dense than the water surrounding them. You can see membranes. So we can now take pictures of cells in a state where if we just thawed them out, they would swim away. It's the real deal, the real state. We call it a native state. And we can take those pictures with an electron microscope, which has high resolution, meaning you can actually see individual proteins. Okay, so let me stop there. So the things that you mentioned that my group has been working on are small improvements in that freezing process. We've worked on ways to get these pictures that we want faster. We've worked on ways to make the pictures a little better. We've developed software to store huge databases of all these images that we've recorded and share them with the world. We've tested different stages in the microscope. So these are really technical details, but a bunch of the effort of my group has been trying to improve the technology and produce tools that help us do this 3D imaging of cells, frozen cells. I'll stop there.
Amazing. So by doing this, you've done work on prokaryotic cytoskeletal filaments. And you found that these were where they weren't expected to be by predictions done by fluorescent light microscopy. So can you tell us why this might be?
Yeah. Okay. Again, I'm going to give you big picture. It's probably best for our listeners. There are three basic kingdoms of life. When you look at, what is related to what, you see these big three kingdoms. One kingdom is called eukaryotes. These are cells with nuclei. Our bodies are made up of eukaryotic cells. Then there's another kingdom of cells called bacteria, and they generally don't have internal specialized compartments. There's exceptions, okay? But generally they don't have as much internal structure and not a nucleus. And then there's another branch of life called archaea, who also don't generally have nuclei or much internal structure. Well, The archaea in the bacteria are very small. Like the cells are often about a micron across, maybe a little more, a micron long. Micron is a millionth, of a meter. And the eukaryotic cells are about 1,000 times bigger normally. So light microscopes can see the shape of a bacterium or an archaeal cell, but they can see large objects inside of eukaryotic cells, because those cells are, like I say, 1,000 times bigger than bacteria. As a result, and eukaryotic cells have fancy shapes and internal structure. Like think about a neuron. It's going to have an axon and dendrites, and it has a special shape. Think about a muscle cell. It has to attach over here, and it has these fibers that cross. And then when it's time to contract, protein machines cause these fibers to come together. So eukaryotic cells have a lot of structure inside. They don't all look the same. And how does the cell give itself a shape and move things around inside? The answer is it has cytoskeletal filaments. The word, cyto means in a cell, skeletal means like a skeleton. Just like we have bones that give us our shape, cells have these protein filaments that are called cytoskeleton filaments that give the cell its shape and give it a structure inside to move things around from here to there and crawl forward, etc. And these were discovered decades before similar structures were discovered in bacteria. And now I'm getting ahead of myself in the story. In the eukaryotic cells, people found microtubules is the name of one of the kinds of cytoskeletal filaments. They saw intermediate filaments, which is the name of another kind. They saw actin filaments, which is the name of another kind, etc. There's other kinds too, but these are the three big families. And using these fluorescent tags I told you about, you could decorate a microtubule with a fluorescent tag, and you could see that it was long and it stretched from here in the cell to over there in the cell. And so people understood that cytoskeletal filaments give eukaryotic cells their shape and allow them to move things around inside. But because bacteria are so small, people couldn't see any internal structure to them. And we hadn't recognized yet which proteins are likely to have the same role as these cytoskeletal filament proteins in eukaryotes. So if you look at a textbook from even 10 years ago and earlier, they'll say that bacteria, they don't have any cytoskeletal filaments. They don't have internal organization. They're basically a bag of enzymes, just a bag. That's an oversimplification. People knew that some had specific shapes, but nobody knew how that happened. Just as we were starting to be able to take lots of pictures of bacterial cells, some groups discovered that there were proteins in bacteria that were a lot like the proteins in eukaryotic cells that make filaments. And some had started to put fluorescent tags on them and see that they seem to form like a streak in the cell. Then we were able to deliver three-dimensional pictures of the insides of these bacterial cells with a resolution where you could actually see protein filaments. And what am I talking about? It would be like a filament that might be 5 or 6 nanometers across, 60 angstroms. So we could actually see them directly. And we saw lots of different kinds in lots of different places.
And we published some papers just showing, hey, we can see this and we can see that, and we don't know what these are. We don't know what protein makes them or what they're doing, but hey, there's a lot of cytoskeletal filaments in bacteria. Another funny thing is that sometimes we didn't see filaments where the people using fluorescence microscopy had seen fluorescent looking filament-like things. And some of those are still puzzling, but another case, it turned out that it was putting the fluorescent tag on the protein was causing it to form a long filament that coil around in a helical shape. But if you don't have the tag on it, it doesn't do that. And so that's another, example of what cryo-EM allowed us to see. We could see cells in a native state, in a state where they could actually live and swim away, and without the fluorescent tags changing anything. And so some of our papers are about identifying the filaments we saw, getting some clues about how they work, and trying to reconcile what we saw by electron microscopy with the results that other people were getting with fluorescence light microscopy. And that work is still going on.
Amazing. And you've also done work and released papers on, well, you published the first structure of an entire flagellar motor. So what adaptations do different species have in terms of these flagella? And yeah, we'll start off with that. My question was more detailed, but we'll do that.
Yeah, no, that's a fun one. So let me give a little bit of intro to flagellar motors. Most bacteria that can swim do it by building a motor, and then extending from that motor a long, skinny filament outside the cell called a flagellum. And what the motor does, it's like an electric drill. It spins, and it causes the flagellum to rotate like the drill bit of a drill. And because the flagellum isn't just a straight rod, it actually has a helical curve to it, like a corkscrew. When it is driven in a rotation to rotate by the flagellar motor, it propels the cell forward. That's how it swims. Now, this flagellar motor is one of the wonders of cell biology. It involves about 40 different proteins, and a lot of the proteins assemble into rings. So there's a lot of copies of these different kinds of proteins, and one ring assembles on top of another on top of another. It has to anchor itself into the cell so that when it turns the drill on, it doesn't rotate. It rotates the flagellum, and the motor stays locked to the cell. So it has to connect with the cell. It has to get through the membranes of the cell without poking a hole in the cell. And so the marvel of that such an awesome machine exists is exciting. How does it assemble? How does it function? How could it have evolved? Has been an interesting topic of discussion. For instance, people wonder, if you had a car engine and a car engine was evolving and it only had half the parts, what good is that? What good is a transmission unless you have an engine? And what good is an engine until you have a radiator to keep it from overheating? So how could this complex machine evolve step by step, with each step giving a fitness advantage that led to its prevalence to provide for the next mutation that might give it. Anyway, one of the results is that the motor seems to be a composite of several other things. It's like sub-machines that each had their own separate function, that eventually came together to have a new and even more glorious function. That looks, essentially sure that happened. Still, though, I sit and in marvel at the number of mutations that would have had to been tried to create all these different proteins and link them together into a flagellar motor, and how many generations would that take? And could each step have actually had a fitness advantage? And the whole thing is just a philosophical wonder for me. So the way we would like to understand this machine is just like you would a car. Let's say no one ever had seen an automobile. And then one day we wake up and there's all these cars around. Well, the first thing we would do is we'd pop open the hoods and we'd look at all the parts. We'd take each one off, we'd squeeze it, we'd see what would bend, what was squishable, what was hard, how do they fit together? Oh, we've got a piston going up and down a cylinder. Oh, and that piston's attached to, something that does this. Okay, so there's a spark plug, and you do experiments on spark plugs to see what they did. And if you're clever, you can figure out how engines work. Well, we've got a similar situation, just it's so small and it's in the side of a bacterium. Here's a flagellar motor. How does it work? So we take it apart and we look at each part and we try to figure out what its shape is and how it fits together with the others. And if we get enough information, we can probably guess how the whole thing works together. That is essentially the field of structural biology. You try to figure out the shape of everything, how it fits together, and understand how everything works by seeing the structure. And the flagellar motors were a challenge because they're so big and complicated. And we can't get into all the technical details of why that makes it a challenge for structural biology. But one of the first things we were able to contribute is the first pictures of intact flagellar motors inside of a cell with the kind of resolution where you could see the individual rings in that machine. So we could see what the whole thing looks like together in the cell. And so that's what we did. And we have since done that for lots of different species. And there's a number of others in our field that have done the same thing. So one of your questions was, what adaptations do we see? Sometimes there's additional rings that give a motor like more strength. You can imagine some bacteria live in dilute water and they want to swim fast. So those cells could have lots of flagella, and flagella could be built for speed. On the other hand, some cells live in, say in your intestine, in mucus, or other, places in the environment where maybe they live in a high viscosity environment and they need a high torque motor that can turn that flagellum slowly but powerfully so it can drill through mucus or something. And sure enough, if you look at cells that live in high viscosity environments, their motors might have different rings, additional rings, bigger rings, and they're built for high power, lower speed. So those are the kinds of adaptations that you see.
Fascinating. So for time purposes, we'll probably move on a bit. But I mean, I'll link papers next to this. I mean, you've done work on chemoreceptor arrays in intact wild type cells that revealed that the receptors were arranged as a hexagonal lattice. You've also done work on enzymatic hyperstructures and carboxysomes, as well as different secretion systems, so like sort of the weapons that bacteria use, and also on HIV. So I guess you pick which topic you want to speak about.
Yeah, let me speak about all of them in just a little bit of philosophy. I think that would be probably the most interesting to your listeners. sometimes science is hypothesis driven, meaning that we have a hypothesis and we design an experiment to test a specific hypothesis. But sometimes science is just looking, it's just observational, and you don't even know what the hypothesis is. I think our work in discovering bacterial cytoskeletal filaments was like that. Honestly, the first ones we discovered, we knew we had on our hands an ability to see inside cells like no one had seen inside cells before. And the first question we asked is, well, what should we look at? And the most popular bacterium for scientific study is E. coli. And we saw that there was 100,000 papers about E. coli by one count at the time. But it was actually thick. So we knew we wouldn't get really good pictures of E. coli. The next most popular bacteria in the literature was Bacillus subtilis. And and there was 10,000 papers about that, but it's even bigger than E. coli. The third most famous one that had been studied a lot was called Caulobacter crescentus, and it was wonderfully skinny. So we put Caulobacter crescentus in the microscope and just had a look, what's inside. And we discovered a whole bunch of, cytoskeletal filaments. And we discovered these chemoreceptor arrays. And we discovered a filament that divides the cell. We saw it for the first time. And so when you take a picture of a cell, you don't know what you're going to see. And honestly, some people have criticized our work, saying that we're not very hypothesis driven. We seem to write papers about lots of different things. That's different than other labs. Other labs might say, we're going to dedicate our work to understanding kinetochores, or we're going to dedicate ourselves to understanding, the ribosome. This is great, and they will pursue it. Or you might pick a disease. We're going to build a lab. We're going to try to understand diabetes, and so that gives you a focus to it. And our papers have been about lots of different objects inside cells, because if you take a picture of a cell in a way that no one's been able to see before, and you see 5 new things, well, the next thing you want to do is try to figure out what those things are and what they're doing. And so it's taken us into becoming cell biology generalists of the cells that we've studied. And that's why we have this long list of things that we publish papers about, because we pursue what we see. And well, I could say more about it. Sometimes we have specific reasons to look at this species or that species. Oftentimes we do. I should be more fair. Oftentimes we have a good reason, but more often than not, once we take the pictures, we end up writing a paper about something else, because we discovered something there that no one knew about that's even more interesting than our purpose for imaging that cell in the first place.
Wonderful. So, we've got a little bit more time. So, can you talk to us about your HIV research? So, you've discovered that the gag lattice was actually a patchwork of loosely connected hexagonal nets. You've also done work on the maturation of it, and that it matches the predictions of the fullerene cone model, and also like the curled sheet model of assembly. Can you talk to us a little bit about this before we then talk about your recent online textbook?
Yeah. And Martha, excuse me for this detail. Are you targeting a 1-hour podcast? Should I split the time with this question and the next? Because I could talk about HIV for a long time.
Yeah, sure, 50-50.
Okay. Then I'll keep it to just a few minutes. And again, try to say something as broadly interesting to your general audience as I can. Let me tell you that, in 1979, nobody had ever heard of HIV.
Mm.
The first cases of AIDS were recognized and kind of classified that this is a new disease. I think it was 1983. And soon thereafter, it was discovered that the cause of AIDS was this virus, the human immunodeficiency virus, HIV. So then scientists try to understand, what is HIV? How does it work? How are we going to block it? It's a beautiful story in that we've learned so much about the HIV virus now. Soon scientists identified it has various layers. It has a membrane around it. And in that membrane, there's called an envelope protein. It's a spike that allows it to penetrate and get inside of your cells. And then inside the membrane, there's another layer of a protein called matrix. And inside there's a layer of protein called capsid that forms a shell around RNA molecules, and the RNA is decorated with nucleocapsid protein, right? So it's this layered object. Well, people didn't know that immediately. We learned that from taking pictures and dissecting it and sequencing the genome and figuring out what proteins are there and solving the structures of those proteins, which means figuring out where how the atoms packed together to form that protein. Some viruses in the world all adopt exactly the same shape, and they're easier to study because they're all identical. But HIV is more like people in that each virus has the same kinds of, features. For people, we've got heads and arms and legs and stuff, but everybody's arms are different lengths and legs are different lengths, and there's a lot of variety in people, even though we have the same parts. That's true of HIV also. The same layers, but each virus is actually unique. And so we had to approach it by taking pictures of individual viruses. And my group was one of the first that got to do that using our method, our cryo-EM method, to make a 3D picture of a single object. And that taught us more about the different layers. Let me just connect it all to the end of the story, which is that I actually spent a couple of years in medical school thinking that I'd be a doctor one day. And I didn't end up finishing it. I fell in love with research science and didn't end up finishing medical school. But when I was in medical school, they taught us that if you were diagnosed with the HIV infection, you had approximately 5 years to live. And that was the prognosis. Through science, through understanding what is inside an HIV, what enzymes are there, how does it get inside cells, and then taking those structures and designing molecules, drugs, that will block it and clog it and disrupt it. Today, people who are infected with HIV can take a cocktail of drugs, and as far as we know, they may live as long as anybody else. There's some side effects, but they may live as long as everybody else, and it's just a tremendous success story of science and understanding structure that leads to medicines that really improve health. And we played a very, very small role in that, in being able to take the first 3D pictures of the viruses in mature state and immature state. And a lot of technical details after that.
That's fascinating. So just to finish off, can you talk to us about your new online textbook, which is called The Atlas of Bacterial and Archaeal Cell Structure, that you recently released, which will be linked next to this podcast, and sort of what were the aims of this?
Yeah, thanks. Yeah, I love this question. Well, the aims were to share with the world all these new pictures that we had. And remember when I started out by explaining this thin section EM, the traditional thin section EM techniques that have filled our cell biology books and our histology books. Some of the histology books are just page after page of an electron microscope picture of a cell with a figure legend telling you what you're looking at. Then the next page is another picture with a figure legend, and it just goes on and on. That's the whole book. And we realized that we should share the new pictures that we obtained of cells in this native state in the same way. At first we thought we'd call it the big picture book. It's just pictures. Then we realized that our pictures were different than the previous ones, in part because they were three-dimensional. We had three-dimensional reconstructions of cells. And if you take a slice out of the cell up here, you might see one interesting thing. But if you took a slice down here, you'd see another interesting thing. And actually, some of the interesting things you can't see in any slice. Imagine a corkscrew. Which slice are you going to show that reveals a corkscrew? So we puzzled, how are we going to do this? We wrote a version of the book that was just two-dimensional pictures that you could print. But we were dissatisfied. And we decided, because all students in the world, or I should say, there's vast access to laptops now, we could share the book in an electronic form, an ebook, that would show cells in 3D. And the way we do that is we make movies where we show them slice by slice all the way through the cell. And we can roll the cell to different angles to highlight the feature that we want to highlight. Then we realized, if we're going to make an ebook instead of a printed book, we could also have animations where we could show what's going on inside the cell by working with animators to make a movie of, okay, this object goes over here and this moves over here, and motors, turn like an electric drill. And we could really explain so much more about what people were looking at with animations. So, and we could make it free. If we had printed this book, it would have cost $300. And frankly, that's prohibitively expensive for lots of, I have a bunch of kids and they're in college and they don't want to buy the textbook. They want the professor to give them a PowerPoint, syllabus with all the material in it, because the textbooks are super expensive, and the whole textbook publishing industry is being revolutionized right now. So we decided to make it an ebook. It can be free. Anyone can look at it who wants. It can also be adaptive. We're going to embed it, the capability of, on the first page, you can say, do I want the light version that I can read in 1 hour? Or do I fall in love with these 3D images of cells? I want to see everything that the Jensen lab has ever produced. Give me the 4-hour version. The thing can adapt. Also, a lot of our pages will show pictures of a cell and highlight features. And then if you want to know more about that one, you can click on a button that says, tell me more about that feature. And it'll go and show you more examples from other species. Or it'll give you an animation that shows you how it works. You can click on words and the definition pops up. It's really an amazingly more rich delivery electronically than we could ever have done with a printed book.
So this is in development. We released version 1. And I'd like to mention that the first author is my colleague, Catherine Ikonomu. She's been working with me in the lab for a number of years, and she's a great writer. And so Catherine and I are the authors, but we show the 3D pictures of 100 or more cells. And these pictures were taken by various people who worked with me in the lab at Caltech over the years. And it's just the first version. We're going to have many more versions in the future with more animations and more movies. That's the other glorious thing about an electronic book. We can update it every other week if we want. And just as soon as we have more content and improved content, we can put it into the book for everyone to see. So we're really excited about it. We're not going to make any money on it. Sometimes, you know, professors make a little bit of money by writing a textbook, and we decided to sacrifice that in order to make it more widely available. We'd rather enjoy the satisfaction of lots of people getting to see the pictures we worked so hard to get than any royalties from a book. So those are the decisions we made. I hope people will check it out and see if they like it.
Amazing. Well, I certainly did, and I really enjoyed looking through it.
Thanks, Martha.
Well, that's all my questions covered. So thank you so much for doing this today. I really appreciate your time. It's been wonderful and really interesting, and I've learned a lot. So thank you so much.
My pleasure. Good luck to you.
And thank you everyone for listening.